Hello everyone, my name is Tibby. I'm from Stoma's Global Product Team. Today I'll be presenting to you Expanding the Boundaries of Spatial Biology, how Stereo-seq version 1.3 maximize insights. For those of you who are not familiar with Stomix or Stereo-seq, we've been around since 2022. And throughout the commercialization journey of Stereo-seq technology, we face countless challenges and obstacles. Nonetheless, we strive to provide exceptional insights for various applications at all levels for your research. We take pride in stating that Stereo-seq is still the highest resolution and largest field of view spatial transcriptomics platform available on the market. We have successfully established over 100 Stereo-seq labs around the world and have facilitated the publication of over 200 research articles using Stereo-seq technology since 2022. So the secret ingredient of the high resolution and the large field of view features of Stereo-seq chips lies in the DNB-seq technology. So the Stereo-seq chips were empowered by the DNB-seq technology. We were generated using the large chip designs of the DNB-seq T10 system and then cut into different sizes for various applications. So here you can see that we have these highly density packed DNA nanoballs on the Stereo-seq chips with the coordinate ID and the molecular ID. After sectioning the sample onto the Stereo-seq chips, the mRNA information will be released and the capturing probes on the chips will be able to capture the RNA released from the tissue. We were able to have 400 pixels or capturing spots per cell, achieving this high resolution and being able to perform true single cell resolution for profiling the different tissue sections with various sizes. We currently have various product solutions ranging from mRNA to total RNA analysis, as well as RNA combined with the low-plex and high-plex proteins. We provide solutions for smaller capture areas, such as ideal for organoids or biopsies, to large capturing chips intended for profiling whole organs or embryos. We also address multi-omics and multi-modalities by ensuring the compatibility with H&E staining for tissue morphology studies and also multiplex IF for the identification of specific protein markers. Our solutions are also tailored to meet various research requirements and to accommodate the different sample preparation methods, such as fresh frozen and FFPE, to optimize the spatial discovery. The Stereo-seq workflow encompasses three different aspects. The transcriptomics profiling by processing either fresh frozen samples or FFPE samples to cDNA libraries, and then sequencing it on DNB-seq platforms, generating the FASTQ file. And after combining with the imaging file, running through the bioinformatic analysis pipelines, we're able to generate a spatial profile in the visualization software. The Stereo-seq version 1.3 solution is a spatial multi-omics solution for fresh frozen samples. It has high capture efficiency, achieving more than 200 gene type counts per cell, and accessible single cell analysis using our proprietary cell segmentation pipeline or utilizing external single cell segmentation methods for an accurate and flexible spatial single cell analysis. Stereo-seq version 1.3 is species agnostics and covering the unbiased whole transcriptome of all species and providing the largest capturing field of view. We offer a standard size chip size up to 2 centimeters square, and it's also available for customization up to 13 centimeter by 13 centimeter. There are multiple application scenarios that can be applied to the Stereo-seq version 1.3 kits and reagent. We have the regular transcriptomics version 1.3 workflow and also the version 1.3 LCD. These two differentiate from each other by either having the regular size chips mounted onto a slide or having a bare chip version that needs to be handled with a pair of forceps and then have the entire workflow running in a uh, chip reaction plate. For the version 1.3 and the LCD, they are compatible with either fluorescent staining of the nuclei or H&E staining to visualize the morphology. And version 1.3 is also compatible with conventional multiplex IF assays. Um, we recommend users to perform an antibody titration and pad experiment before proceeding to the actual transcriptomics workflow. And after obtaining the imaging, the samples will go through tissue permeabilization to release the RNA and be captured by the capturing probes on the chips, and then perform reverse transcription and then cDNA release to generate the cDNA library, and then will be amplified and purified before constructing the final Stereo-seq library before sequencing. Stereo-seq version 1.3 is optimized for fresh frozen samples. So there are certain recommendations and requirements listed here. For example, for the tissue section thickness, we're required to have it at 10 microns to optimize the mRNA being released onto the chips, and the recommended ring value for the RNA quality is equal or greater than 4 for the smaller size and the regular size chips, and greater or equal to 7 for the large chip designs. And in order to optimize the downstream analysis, the tissue should not exceed 80% of the area coverage of the chip. And currently, the Stereo-seq version 1.3 IMIF solution is only available for the 1 centimeter by 1 centimeter chip sizes. So here is a general overview of the version 1.3 product solution. It's targeting the 3' RNA, profiling the unbiased whole transcriptome of fresh frozen samples. It's applicable for various applications such as neuroscience, ontogenesis, organ research, or cancer and disease models. The kit that will be used for the workflow includes a permeabilization set. This is for determining the optimal permeabilization time. And after determine the permeabilization time for your sample, you can proceed to the transcriptomics workflow using the Stereo-seq transcriptomics set to process your sample to the cDNA. And after that, you will use the 16 barcode library preparation kit to prepare the cDNA library for before sequencing. And for version 1.3 libraries can be sequenced using the Stereo-seq virtualization reagent set for the G100 platform or the T7 platform. For the version 1.3 multiplex F protocols, and this is still targeting the whole transcriptome, and it's suitable for the standard size samples. Besides the regular permeabilization set and the transcriptomic set that will be used for the version 1.3 workflow, additional antibodies need to purchase separately, and we recommend users to perform antibody titration, and the optimal antibodies to be combined will be about 3 to 5 antibodies in a group. So we recommend to choose the optimal concentration of each of the antibody and then perform a pilot experiment with all of the antibodies you've chosen to determine that all of the antibodies are can be clearly visualized under the microscope. There's no photo bleaching and no interfering of the secondary fluorophore signals. And then you can proceed to the regular Stereo-seq transcriptomics experiment using the Stereo-seq transcriptomics set. For the Stereo-seq transcriptomics LCD solution for version 1.3, the workflow is similar to the regular size chips. there are three different sizes can be applied: the 1 centimeter by 2 centimeter, 2 centimeter by 2 centimeters, and 2 centimeter by 3 centimeters. And same as the version 1.3 regular chip sizes, you need to perform the permeabilization time point determination using the permeabilization set. And then after determining the optimal permeabilization time, you can proceed into the transcriptomics workflow using the transcriptomics set for each chip sizes. To support the spatial transcriptomics analysis, we have this complete software suite composed of two softwares with three major components. So the image processing, the reads processing and quantification, and then the visualization. So there are two softwares that we use. One is the Stereo-seq analysis workflow, the SAW pipeline. This is used to to process the raw FASTQ file and image data, and also to perform alignment and spatial mapping and generating the gene expression matrix along with the clustering results. And for the stereo map software, a will be used at the image processing step for image quality control and some image manipulation and adjustment. And later on, after running the data in the SAW pipeline, the gene expression matrix can be visualized in stereo map as well to look at the marker gene expression and then also the spatial clustering results. So the importance of delivering the Stereo-seq technology and product solutions to all of you is to focus on enabling the true single-cell discoveries and for your research projects, since this is always the best way to fully utilize the spatial fidelity and also to uncover biological insights that most accurately represent the disease and the cell state. So we obtain the spatial single-cell level gene expression profile by projecting the high-resolution RNA data onto cell-segmented microscopic images. And we utilize this software called CellBin. This is an integrated image facilitated cell segmentation framework for high-resolution spatial transcriptomics data to ensure accuracy. So you can see here with cell-based segmentation, we can perform interactive exploration of the immune microenvironment, revealing the spatial patterns of immune cell distribution and gene expression at the single cell resolution. There are various cell segmentation tools available out there. Um, So the SAW pipeline and also our visualization software tools support not only our proprietary cell segmentation, the results generated from our proprietary cell segmentation algorithm, but we also support the results to be inputted back into the SAW pipeline using a third-party cell segmentation tool, and then visualize it using the serial map software. So you can decide on what self-segmentation tools fit or works the best for your research project or your sample type. With Stereo-seq, we now can provide more insights with less effort, generating a richer data and providing a better solution for advanced high-resolution spatial transcriptomics research. In this example we're showing here, this is a semi-section of a mouse brain. We can profile over 100,000 cells on in a single experiment, and each with nearly 1,000 gene type counts per cell. Here is a remarkable data demonstrating that with uh, the analyzation of the gene expression profile through the cell-beam analysis, we're able to review the intricate spatial cellular structures of the mouse intestine. So the Stereo-seq version 1.3 enables the precise tissue stratification through detailed spatial single-cell analysis, and this is providing a well-defined tissue stratification, being able to look at the different layers of the cells within very intricate tissues. And the arrangement of the cell subtypes and the expression of the marker genes, they align really well with distribution patterns of related cells. In this case, here we're demonstrating here that the spatial single-cell information can review pathological changes in cancer samples. So here, this patient has been diagnosed with human esophageal cancer, specifically the squamous cell carcinoma, ESSC. This is a common esophageal cancer accounting for the majority of esophageal cancer types. So the patient was diagnosed with ESSC on the premises of clear observation of tumor cells infiltrating the basal layers of the esophageal tissue. So the squamous epithelial cells were observed to be surrounded by fibroblasts, indicating invasive characteristics. We then delve further into the molecular changes of the sample by examining the spatial single-cell results alongside histological information from the same tissue section. So after profiling this sample using Stereo-seq workflow, we were able to uncover and recognize new insights. Here, we can precisely annotate the cells and gather spatial cellular data, which uncovered a rare cell type, the glandular epithelium within the tumor area. We discovered that the glandular epithelium was situated beneath the squamous epithelium of the esophageal mucosa, displaying a tubular structure and invasive traits in the muscle layer. So this has been very insightful and providing results that match the histology validated image. For instance, the spindle-shaped fibroblasts were seen in the H&E image near the rectangular squamous epithelium marked by the green arrow. So the Stereo-seq single-cell annotation has successfully confirmed this finding and then verifying that the fibroblasts were located directly adjacent to the squamous cell. In our examination of the spatial molecular data exhibited cellular features that harmonize with the histological and diagnostic attributes. Besides the regular transcriptomics applications for the Stereo-seq version 1.3, we are also compatible with MIF staining, which enables the identification of biological niches and high-resolution molecular mapping. Here, we're demonstrating that we were able to profile the mouse brain and then um, overlay the DAPI and also the antibody stain image on top of the gene expression profile, and then to be able to visualize the protein distribution pattern with specific tissue region of interest after performing the spatial clustering. So Stereo-seq version 1.3 LCD solution provides another layer of or level up the solutions that we can offer by providing various chip sizes to accommodate different tissue sizes. So for the standard size product, we have the smaller size chips and also the regular size chips, which offers a more cost-effective option for smaller samples. And we also have this Stereo-seq LCD standardized product for 3 different sizes. That's ideal for profiling large tissue types and also for the entire organ. The importance of having a large capturing FOV is the ability to explore multi-organs simultaneously on one experiment. Here we're showing that there's the mouse embryo at E18.5 days. Uh, the multiple organs can be captured and explored in high resolution, all in a single experiment. And this type of organ level profiling is especially valuable for developmental biology and system level studies, which can enable you to analyze the tissue heterogeneity across organs, identify organ specific gene expression patterns, and study spatial relationships between developing structures. And in this data, we were able to look closely at the bone development within uh, various stages of the mouse embryonic development stages. So here we're looking at the stage-specific ossification across skeletal structures. You can see here that the mandible, the vertebrae, the pelvis, the ribs, and the long bones in the mouse embryo, they primarily underwent endochondral ossification, which is also the fundamental process underlying fracture healing. In this process, chondrocytes first proliferate, enlarge, and eventually undergo apoptosis, after which they are replaced by bone tissue. So here we can clearly observe the stage specific ossification status across different bones. On the lower layer, we're shown here are the pelvis and the ribs and H&E staining, and the clustering results demonstrated that the pelvis and the ribs are still entirely composed of cartilage at this stage at E18.5 days of the mouse embryonic development timeline. And the long bones and the vertebrae here are in active transition. They're undergoing endochondral replacement, with some of the cartilages has been replaced by bone tissues. And the mandible here has already completed most of its ossification, suggesting that the mandible development precedes that of other skeletal elements, which having the entire embryo to be able to process in large field of view enable the precise localization of key structures and cell types involved in bone development, such as the hypertrophic chondrocytes and the trabecular bone. For Stereo-seq version 1.3, we have validated across multiple species and sample types, and there are more that are being validated and being tested with internally or with our existing collaborators. So how to choose the right platform for you if your project is mainly focused on transcriptomics, and if you have different sizes, then you can decide first on what chip sizes that you wanted to choose. Is your sample small, or if you wanted to mount multiple small samples on an entire large chip, we have different solutions for you. And for the Stereo-seq version 1.3, it's compatible with either the SSD staining to stain the nucleus or H&E staining to stain for the tissue morphology confirmation. And for multi-omic solution, we have the Stereo-seq transcriptomic version 1.3 with the MIF protocol available for the 1 centimeter by 1 centimeter chip sizes. And there's another high-plex protein profiling solution available, which is called Stereo-seq, focusing on uh, uh, capturing the antibody, the high-plex antibody with the RNA information at the same time, and then sequence it all together. So the Stereo-seq introduction will be included in our upcoming webinar, so please stay tuned for that. And we will go a little more in detail of what are the differences between the version 1.3 MIF and Stereo-seq. They each have their merits and can benefit your research projects by designing it carefully with our solutions. If you're interested to explore more of how to perform the experiment, how you can analyze the data, please go to our website. We have all of the resources there, including the user guidance and the experimental preparation checklist to jumpstart your project, and also for operation videos on how to handle FFPE sections and how to section the fresh frozen tissues. There are operation videos available for you as well. And please feel free to check out our demo data on the website and also to read through our blogs. Thank you again for joining our webinar, and we hope to see you in our next webinar.
Okay, thanks for TV's sharing. And today I will walk you through a full workflow of a Stereo-seq project and show you how exactly these powerful tools can be used in your really research. And to ensuring the connection, I will close my video sharing. Stereo-seq has attracted lots of attentions from the scientific communities, and over 160 publications has been published by July 2025, and over 100 of those papers have an impact factor over 10. It's not all about publications. Stereo-seq has successfully applied for in several research areas like the organ development, tissue regeneration, oncology, neuroscience, and plant biology. So, as we mentioned before, we now offering four solutions. So, with these available solutions, you might think this sounds great, but how exactly I start a real Stereo-seq project, and how should I prepare to make this run smoothly? So, those are the questions we are going to answer today. To make it real, we will use a demo case called sleep deprivation to show you how to do the project planning, sample preparation, experiment, data processing, and with the metrics, we will show you how to do the analyzing like the cell annotation, differential gene expression, cell communication, and other analyzing work. So let's talk about the sleep deprivation background. Sleep is quite important for everyone. It helps us to imbalance our brain, control our emotions, and improve our memories. So if we don't have enough sleep, we are not only will feel tired, our cognitive functions will decline, and our learning and memory will impaired. So previous study has shown that mice under sleep deprivation has the same symptom as human being, but there are limitations. Current research only focused on 10 to 100 target genes, and they usually focus on one area of the mouse brain. But now with the capability of Stereo-seq, we can study the whole transcriptomics of multiple regions of your slice of mouse brain. Let's see how we start our sleep deprivation project and how to do the sample preparation. In this project, we have 16 mice and we divided them into two groups, the control group and the experiment group. Because mice are nocturnal animals, which means they usually sleep under light. So at the first 5 days, we keep them in 12-hour light and 12-hour dark cycles, and to let them get used to the stress of the later process. We kept locking the cage for 5 minutes at the beginning of the light circle. And at the day 6, we let the control group keep sleeping for 5 hours. But for the experiment group, we kept tapping the cage to let the mice keep waking for 5 hours. And after 5 hours, we dissect them immediately and take out the brain and embed it with the OCT medium. So if you have questions about how to do the sample embedding, you can look on our website. There are protocols to show you how to do the sample embedding step by step. And we only take 6 out of the 16 samples because some tissues are damaged while we are doing the sample embedding. So there is a tip for you. Please always prepare the extra samples in case of the emergency or small areas of the progressing. After we get the sample block, now we need to do the Stereo-seq experiment. There are three experiment tissue kits for the whole process. we will get the perfect permeabilization time with the permeabilization set. And we will use this permeabilization time in the transcriptomic experiment. After we get the cDNA with the spatial information from the transcriptomic experiment, then we will use a library preparation kit to transfer the cDNA into the sequencing library. And then we could use a next generation sequencing machine to sequencing all the library. And there are several steps in this experiment processing, like the tissue slice, staining, imaging, and RNA capturing. So our colleague will show a detailed demonstration of this part of work and tomorrow. After we get the sequencing date, we will use a software to do the sample processing. We call it SAW. This is a user-friendly software. You can only need to download it from our website and then unzip it. Then you can start running it without any extra installation step. So this you can see here, this is a command line with SAW count. So with this command line, we input all the materials like the reference, the sequencing date, the chip mask, and the image files, which generated from the image step of the wet lab work. And after several hours running, we can get the output of the SAW. The matrix was included in the output files, and then we will need to use a stereo map to visualize our data. I will quickly show you how it look like with the stereo map 4. So this is the uh, so you can see all the details with stereo map like this. And okay, our colleagues tomorrow will show you a very detailed introduction about this part. So there is a report in the SAW output files, and all the informations about your experiment of your samples will include in this report. And I summarized all the informations of our 6 samples in this project. You can see here we got 110 to 140,000 cells per slice, and with 1.6 to 1.7 billion rays, we can get nearly 700 to 1,000 median genes per cells. So this is a quite rich data set. We can do a lot of analyzing work with this data set. Before we actually doing the analyzing, I'd like to introduce Python package to you, StereoPy. The articles about these tools lately published, and StereoPy can provide plenty of functions, and like the PCA, clustering, cell annotation, cell-to-cell communication, and multiple sample analyzing, even for the 3D structure constructions. So we put our data expression matrix in the GIF files. So these GIF files can help you to transfer our samples into different bin size, which means you can transfer those expressions into any size you want. And those are the script you can use for the data reading. And we are not only construct ourselves with the StereoPy analyzing work. You can also use the StereoPy to transfer the GIF files into other software into other formats like the H5AD and RDS, and then you can later use the other software like the ScanPy or Thread to do the analyzing work. So let's look at the cell annotation. We used a reference-based annotation software to transfer 87 cellular subtype labels into one sample or one brain slice. You can see here the isocortex annotation. The cell groups shows the similar structures like the anatomic structures of the mouse brain. And we are Stereo-seq data not only can use the reference-based annotation, we can also use the cluster and marker to annotate the data set. Like some researchers may not have qualified single cell data set. So let's look deeper into the UMAP of those cluster. You can see cell cluster in two distinct clusters of the hippocampal, and they consist with the anatomic structures of the hippocampus. And as for the marker genes, steroidate share the same marker genes as the alien brain single cell atlas. Now let's look at the differential gene expression. We use StereoPy to do the cell differential gene expression analyzing. So we divided the cell into several brain areas. For the isocortex, we can see here genes are enriched in the circadian regulation. So it makes sense because all the mice under the sleep deprivation, um, the circadian rhythm was disrupted. And let's look at the hippocampus. We can see the regulated genes are enriched into the synapse organization and signaling. So actually, the hippocampus are related to the cognition and memory formation. So this shows that sleep deprivation may affect the cognition and neural interaction. And let's look deeper into the DGs in the hippocampus. We specifically focused on one gene, LRTM4. We can see here the LRTM4 was downregulated in the experiment group. Even we can see the difference with the spatial plotting. So a previous study showed that the density of the dendritic spines reduced with the LRTM4 lockout. And for the aged rats, the LRTM4 protein expression decreased. And in the Barnes maze test, the aged rats took more times in this test. The whole story shows that um, the LRTM4 downregulation shows that sleep deprivation may impair the mice cognitive function. And we want to look deeper into the memory formation. As we can see, the hippocampal is the structure where the memory formed, the and there is a tree circuit. We call the tree. The information was collected from our eyes and ears, then transferred to DG area, then to the CA1 cells. So we want to look deeper into whether the sleep deprivation affected the circuit. So we use the cell chat, and as this, we can look at this picture. The blue line indicated the decreased interaction between the cell groups. So we can see here the DG glued to the CA3 glued neural and then to the CA1 neurals. All the interaction between these three groups are decreased. And when we look deeper into the ligation and receptor interactions, we can also see the same results. You can see clearly the interaction in the sleep deprivation group. The signaling were downregulated. So this means that sleep deprivation may affect the mice memory formation through the ligation and receptor interactions. I believe there are still plenty rooms for us to dig, but and we will upload all our demo case and the results into our website, as well as the scripts. So if you are going to start a new Stereo-seq project, you can download all the data and to run first to make sure you could run your project smoothly. And thanks for your listening.